students. I shall take up your subject Herbal Drug Technology, code BP603T, and I shall start with the first chapter, Herbs as Raw Material. Herbs are used in traditional system of medicine since time immemorial to alleviate human illness for the maintenance of general health. हम सबके घरों में हम शुरू से since ages we have been using plants and the materials to cure the बीमारी को कम करने के लिए. Plants or herbs as a medicinal product is nothing new for us. 80% of world population of the developing countries still rely on the crude drugs and the folklore medicines for their primary healthcare needs. It's our traditional belief, yes, हम इस जड़ी बूटी से ही ठीक होते हैं. That is our folklore medicine. And you know that more than 40% of that lies with the rich heritage of the India and China. There is a need to adopt systematic methods for their selection, cultivation, collection, processing so that हम उसकी quality को, purity को, safety को और potency को ensure कर सकें. That is why we need to study this subject called Herbal Drug Technology. Before we start, we have to get acquainted with a few terms. कुछ terms ऐसी हैं जो हमें अच्छे से समझने की ज़रूरत है. Say for example, herb. What is herb? Herb has been derived from the Latin word called herba, or you can say an old French word called herbe. It means any part of the plant which can be a fruit, a stem, a leaf, seed, flower or just a petal used for the flavoring, food, medicine or perfume. Even the stamens have been included. आपने देखा होगा केसर, तो केसर भी एक herb ही है जो कि इन parts में नहीं है, but stamen जो कि flower का एक part है. That is also one of the components. Crude material derived from plant, fungi, algae, lichen, roots, rhizomes and other plant parts which can be either entire, आप उसको as a whole use करते हैं, fragmented, उसका कुछ हिस्सा use करते हैं, या फिर उसका powder करके use होते हैं. So that all constitutes the word herb. Now, what is crude herb? Crude मतलब जो कि nature में as it is exist करता है. हमने उसको कोई treatment नहीं दिया, किसी process से वो pass नहीं हुआ. That is a crude herb. So let us go by the definition on this slide now. Whole, fragmented or cut plants or its part of the plant or maybe algae, fungi and lichen which is not processed. आपने उसको processing नहीं दिया है. Say what is shown here in the picture. Acacia senegal. Now this is the gum acacia which oozes out from the bark of this tree. Then any other plant part which undergo only the process of say collection and drying. अगर किसी process से वो pass हुआ है, so that is just collection and drying. So here is shown dried bark of Cinchona officinalis. This crude herb can be fresh or it can be dried. It is basically including the exudate from the plants as well. The other examples of crude drug are given in the table here. The number one is fennel. Fennel जो सौंफ हम घर में अक्सर यूज़ करते हैं. That is dried ripe fruits of Feniculum vulgare, family Umbelliferae. Then belladonna. It consists of dried leaf and flowering tops of the source Atropa belladonna, family Solanaceae. Then मिर्च which is a common name जो हमारी spices में condiments में use होता है. This is dried ripe fruit of Capsicum annum, again the family Solanaceae. Then linseed, this is another example of the crude drugs. This is consisting of seed of flowers of Linum usitatissimum, family Linaceae. So these are the just 4 examples of the crude drugs, and whatever you're going to study in the subject of herbal drug technology and maybe pharmacognosy in the next classes, they all undercome the crude drugs. Now, after originating or collecting the plant part which is a crude, we have to process it so that it becomes usable. It becomes in the form which is consumer friendly. So that is what it is called as processed herb. You are giving some processing to it. It has to pass through processes. Fine. So let us study what does it mean. It is preparation obtained by subjecting the herb to treatments such as extraction. आपने extract किया है. You have taken the plant part. You have extracted out its active constituent जो आपको चाहिए है API वो आपने उसमें से निकाला है. और you have subjected it to the distillation. आपने उसका purification किया है. You have expressed it, expression where the oils are generally expressed from the flower's petals. Then fractionation, you have divided into different fractions or the strata or one part which you need. Then purification, you have purified it. Concentration, when the content of the API is lesser, you subject it to the concentration process. And partial or full fermentation. So these are the various techniques for the processing of the herb. These are extraction, distillation, expression, fractionation, purification, concentration and partial or full fermentation. So what do we get out of these crude extract which are processed now? We do get tinctures. You might have heard the tinctures, ginger tincture, extracts, essential oils which you generally extract in your labs, fatty oils, extracted exudates, gums, gum acacia, gum arabic, oleoresin which is a combination of the resin and the oil जैसे कि balsam and liquid extract. So these all are the examples of the processed herb. Now what is herbal medicine? You know what is medicine? Medicine is something which is having a therapeutic effect, which is basically purported to cure some disease, to give some therapy. Now, when this therapy comes from the plant, it is called as herbal medicine. So, what are these? Let us go by the definition on the slide now. These are finished labeled medicinal product that contain active ingredient. They can be either crude or processed, which we did in the previous slides. They may include juices, gums, volatile oils, fixed oil or other substances from the plants. Medicine possessing crude drug mixed with chemically distinct substances including phytoconstituents are not termed as herbal medicines. You need to note down this point that the possessing which are having isolated phytoconstituent are not termed as herbal medicines. Herbal medicinal product. When we are talking that a medicine has to combine with other additives to make a say dosage form or to make a patient friendly form, that becomes herbal medicinal product. Now these are the products produced exclusively from the herbs used in medicinal purposes. They are defined as medicinal product exclusively containing one or more than one active ingredients of the herbal origin. They play an important contribution in any country's gross domestic product. It includes various herbal formulation like syrup, mixtures and tablets. Herbal drug preparation. Now these terms are quite overlapping, there is just a slight difference between them. Herbal drug preparations are again the one which is obtained from the herbal drug only. They again are under processing which has been done. They are prepared by the various techniques such as extraction. In extraction, we take out the active constituent of the plant into our solvent in which it is soluble. Say we carry out extraction in sometimes say in ethanol, right? Or we take the extraction in the water. तो जिस भी solvent में plant के constituent soluble हैं, हम उसे as an extraction solvent लेते हैं. Then infusion. In infusion, generally we add boiling water to get the content. Then we have decoction. Decoction में हम प्लांट को बहुत देर तक रखते हैं एंड वी कीप इट बॉइलिंग अगेन विद द सॉल्वेंट सो दैट उसके एक्टिव कॉन्स्टिट्यूएंट्स बाहर आ जाएं, लीच हो जाएं. ठीक है. मैसेरेशन, मैसेरेशन एक टेक्निक है जिसमें कि हम प्लांट के अगेन जो भी कॉन्स्टिट्यूएंट्स हैं उनको एक्सप्रेस करते हैं सॉल्वेंट में. मैसेरेशन कैन बी वन टाइम और इट कैन बी मल्टीपल टाइम्स. अगर एक बार में एक्सट्रैक्ट नहीं आते हैं हम फिर से मैसेरेशन करते हैं. देन वी हैव डिस्टिलेशन, then we have expression, fractionation, purification, concentration and fermentation. These all are the techniques which are used to make a crude drug into a processed drug, and these are the techniques which are helpful to make the herbal drug preparation. Now these herbal drug preparations are available in the market in the various forms such as powders, extracts, tinctures, fixed oils, volatile oils, resins, gums, etc. These herbal drug contain various constituents. Sometimes phytoconstituents are isolated and formulated in appropriate dosage forms in place of the entire parts. That is what I said to you, that you have to convert into dosage form which you can take up. Now, where do these herbs come from? What are the various sources of the herbs, right? The herbs can be obtained from three sources. Number one, the wild source, wild, which exists as in nature. So, they are existing in the nature as it is, say from the forest. Then, cultivated source, where a cultivator or farmer grows that medicinal product to get the plant part. Or the third way is by using modern scientific techniques like tissue culture, polyploidy, mutation, hybridization, genetic engineering and germplasm. So these are basically the biotechnological techniques by which we are enhancing the yield of the plant or increasing any of the constituent of that plant. So these are the three sources of herb: wild sources, cultivated sources and the modern scientific biotechnological sources. So let us understand the difference between cultivated sources and the wild sources. Cultivated sources, here we can ensure the quality and the purity of the product. Since we are growing these sources, we are providing them essential nutrients, we are taking control of the pest, we are taking care of everything, we are nurturing them. So the purity and the quality of the plant product can be ensured. Other feature is that we can also apply modern scientific technique to this source. Besides that, it leads to industrialization and has given right to several cottage and small scale industries. The plants which we are growing at our own will, by our own methods, only then can be transferred to the industries, right? Then the rapid growth of phytopharmaceutical and perfumery and allied industries is possible due to cultivation of the medicinal and aromatic plants. On the other hand, the wild sources which I told you earlier, the one which exists in the nature itself, the one which exists in the forest, the one which come up like weed, the one exists in the rainforest, so they are occurring in the nature at their own. Obviously, they have number one advantage, that they are economical. You are not putting any sort of efforts to them. They are less time consuming because they are growing in the nature at their own. And obviously, the disadvantage is that the quality cannot be predicted due to the various environmental changes. If there is less rainfall, if there is higher rainfall, so you are not taking care of the quality. It is happening whatever is happening in the nature at its own. There is no uniformity, like you cannot have a control on the yield and the growth and the other characteristics of these wild sources. They are widely distributed in the different locations, and there are chances of adulteration and substitution as well. They may lead to depletion of the raw material. If the same plant is grown in the same soil again and again, it is going to deplete the soil of its nutrients. So that is going to be condition which is going to happen, depletion of raw material. It cannot fulfill the demand when required. If you need a high demand, you cannot trust over that. So this is the major striking differences between the cultivated and the wild sources. Now, the technique called plant tissue culture comes under the third category, the biotechnological sources which yield the plant in the required concentration with the required active constituents. Plant tissue culture is a technique which is an in vitro cultivation of the plant cell or tissue under aseptic and controlled environmental conditions in liquid or semi-solid well-defined nutrient medium for the production of primary and secondary metabolites to regenerate the plant. What do we do here is, we take a plant cell or a tissue, we grow it outside the body of the cell. So that is a term called in vitro. Then you are providing all the necessary growth factors to it. Here you can see well-defined nutrient medium. You are providing the aseptic control. There is no source of microbial contamination, there is no accommodation of the pest over there. You are taking strictly control of all those things, and then you are growing healthy plants. This technique affords alternative solution to the problem arising due to current rate of the extinctions of the medicinal plants. There are many plants which have been extincting now, and now we can grow them in by this technique called plant tissue culture in our tissue culture labs. They require a well equipped culture. Obviously, this technique is not that very simple. You have to have a assembly, you have to have a nutrient medium and a cultural laboratory for this. Basically, it involves two processes, and the two techniques are involved here. Number one, preparation of the nutrient media. Now, the nutrient media which contains or which provides all the organic and the inorganic salt which are required for the plant growth, say nitrogen, phosphorus, potassium, whatever is required for the plants, we provide it all externally. Besides that, we provide plant growth hormones as well as amino acids which are necessary for the growth. In addition to that, the sterilization of the X-plant, the plant part which we have taken as a seed, as an origin of this growth, say plant tissue or plant cell, that is an X-plant. Now, that X-plants which has been taken has to be sterilized, right? And we require a proper glassware and the other accessories which are required for the inoculation and incubation. आप एक strictly controlled aseptic environment में plant के growth regulators ऐड कर रहे हो, plant के nutrients ऐड कर रहे हो और फिर उस plant को grow कर रहे हो. So there are no chances of any sort of contamination. This is what plant tissue culture is. This is one of the examples. It is basically a research work done by a group here. We can have different stages of the plant tissue culture here. You can see in the form of the different notations given A, B and so on. So first of all, let us have, this is a plant called Sanguinea and have a look on the figure A. Now this figure A is sterile indecent capsules which are soaked in sodium hydroxide. Then B, this is soaked in the hot water. Now these seeds have germinated. If you can have a look here, these seeds have germinated. These are germinated seeds. Now they have converted into seedlings. Figure D shows you the seedlings. After this, the seedling has shown the growth of adventitious shoot system. In the figure E, we can see here, this is at the stage of 20 days and this is at the stage of the 40 days. Now, this is the clustered shoot which we can see again having the roots as well. Now this is seen when you see the plant tissue cabinet. From the bottom you can see the roots, and finally you have the plants which are ready for the transplantation to the either the plant pot or to the field. So these are the finally the result which we get. So these are the different stages, and this is finally ready after 30 days. So this is the diagrammatic view or the pictorial view that how the plant tissue culture is done. Another technique is called polyploidy. Polyploidy is nothing much but it is increase in the number of chromosomes in certain tissue or entire plant in multiples of basic or haploid number. It is caused by cell generation, physical agents like X-rays, centrifugation, temperature, shock, and the chemical agents like colchicine. Now, the advantages of the polyploidy. This leads to the healthier, stronger, and larger than the diploid counterparts. Polyploidy plant contains larger flowers, pollen grain, and stomata. The phytoconstituent of some plants can be increased than their counterpart diploid plants, but it has disadvantages also. The effect of polyploidy is not generally predictable, because what you are doing here, you are making the change in the genes. See, these are the genes and these are the chromosomes. So you are attempting to act on those genes which you desire. The gene which is responsible for the production of secondary plant metabolites. Say, if you want that there should be increase in the content of the alkaloids, or if you want that the tannins should be higher than the normal production, then you can go for these techniques. So, you are making the genetic changes, you are making the genetic manipulations so that you can get that result. Hence, because genes are not always something which you can rely upon. You cannot trust that whatever changes you are going to make, it is going to happen, right? This is actually happening. In the COVID scenario, the next genes are coming and you are getting another strain, fine. So, the effect of polyploidy is not predictable. The other disadvantage is that the epigenetic instability that results in transgressive gene regulation, again something you cannot have a control. Another biotechnological technique which we need to study is hybridization. Hybridization is what you have always heard of, that you get a hybrid tomato, right? आपने देखा होगा आजकल टमाटो सारे के सारे रेड होते हैं. इज़ इट पॉसिबल कि सबके अंदर ल्यूटिन इतना ज़्यादा हो कि सारे टमाटो रेड होते हैं? नो. This has come by the technique of hybridization. Then you might have seen the seedless watermelon. राइट, वाटरमेलन विच इज़ सीडलेस. वाटरमेलन के अंदर सीड ही नहीं है. नहीं तो जब समर्स में वाटरमेलन एंजॉय करते हैं आप उसके सीड निकाल कर खाते हैं, है ना. अभी क्या है, सीड है ही नहीं. तो वेयर डज इट कम फ्रॉम? सो दिस कम्स फ्रॉम द टेक्निक ऑफ हाइब्रिडाइजेशन. हाइब्रिडाइजेशन हैज़ बीन यूज सिंस ईयर्स टू गेट दी प्रोडक्ट यील्ड. वीट में हमने यूज़ किया, हमने इसको एग्रीकल्चरल प्लांट्स में बहुत ज़्यादा एक्सप्लॉइट किया है सो दैट वी कैन गेट द right amount and the right content of the active constituents for the population. So, what is hybridization? It is a natural or artificial method in which there occur a union or crossing of two genetically different plants. आप दो plants का mixture करते हैं so that you get what you want, right? The final produced plant is known as hybrid. Hybridization does not modify the genetic content of the organism. However, it produces new combination of the genes, right? Advantages, obviously it offers that the desirable characters of two different plants can be obtained in a single hybrid plant. Earlier, we could not eat cucumber throughout the year, है ना. सारा साल खीरा नहीं मिलता है. It is a basically vegetable of the summer season, but now you get throughout the year. Why? Because you have that hybrid variety which can sustain in the low temperature also. It can be also grown at the low temperature as well. Similarly, there are large number of fruits and vegetables which are available throughout the season, and this is all because of the hybridization. You have finally come to a breed which is having the characteristic of both the plants which you have taken as an individual participant in the hybridization. The other advantage is that the phytoconstituents quantity can be increased. You know, the phytoconstituent quantities are very less. If you take some plant, say अगर आप जीरा लेते हैं, cumin, or you take the clove or any other which contains the say volatile oil, तो जब आप इन प्लांट्स की एक्सट्रैक्शन करते हैं, वॉलेटाइल ऑयल का कंटेंट बहुत कम होता है. इट इज़ मे बी 1% or 2%, maximum 3%. It is lesser than 1% generally. So one, you want to increase that volatile oil content, so that is a phytoconstituent basically. So you can apply this technique called hybridization.